I wanted to kill you, hopefully I'm smart enough to hide my DNA from the police. Deoxyribonucleic acid carries the genetic information of all organisms, being unique to each one. The smallest and most basic form is called a nucleotide, coming in 4 different types: A, T, C, and G. They're composed of a nitrogenous base, a phosphate group, and, very obviously, deoxyribose sugar. The phosphate groups connect to form these long-ass polymer chains. The nitrogenous base is paired due to hydrogen bonding to form a twisted ladder structure known as the double helix. This is jam-packed into a chromosome. As mentioned, it's a long-ass structure known as a biomacromolecule. And there's a reason why us biologists don't use chemical formulae. Anyway, what if I were to see or touch it? Well, it is indeed possible. You could extract DNA from any organism, just don't grind up your pets, guys. Let's try out this experiment. I'm going to be extracting DNA from strawberries, and you could use many alternatives. Some of you like me would have done it in school already. Here's what we need. 15 grams of fruit, 25 ml of DNA extraction solution, which is just a mixture of water, detergent and salt, approximately 15 ml of cold alcohol, a Ziploc bag to hide the evidence, a big and small beaker, a straining cloth, and a wooden skewer. Alright, first we put all the strawberries into a bag and smash. This exposes as many cells as possible for the next step. Now we need a secret solution, the DNA extractor lysis solution. 2 teaspoons soap, it's not what it looks like, guys, come on.
Hey yo!
Half teaspoon salt and half a cup of water. Pour the lysis solution into the bag of strawberries. It may not look like much is happening here, but it definitely is. You see, this is a plant cell. Each organelle is membrane bound and that protects these organelles. What we need to do is break apart the phospholipid bilayer in the cell wall and nuclear membrane. Look here. 1 polar head, 2 nonpolar tails, and this is soap. The molecules are similar enough that the soap can slip in and tear them apart. And this exposes the treasures of the nucleus, the DNA. The salt would break up the protein chains binding the nucleic acids. After filtering this into a beaker, we'll transfer this mess into a test tube to make things easier to watch. And now the cold alcohol goes in. The DNA begins to separate. A white, sticky, stringy substance precipitates out of the red liquid. After drooling over this, I use the skewer to scoop it up. That's just stuff, guys. It's crazy though, how easy this seemed and yet how extensive the field of genetics and molecular biology is. Everything lies within the small things in life. Keep that in mind. Before the FBI catches up to me, be sure to share this experiment with your schoolmates and don't forget to subscribe for biological experiments and explanations. This is A.B.S.E signing out. Take care and... FBI, open up! Uh-oh.